Medical Laboratory Hematology 2, 521. Okay, so in this section of the course, Medical Laboratory Hematology 2, we'll be looking at the aspects we'll be looking at here specifically is molecular diagnostics in hematology. We'll be looking at molecular diagnosis in hematology. Now, course content include, we'll look at the DNA structure and function, looking at also introduction to DNA analysis, molecular diagnostics in hematology, application of molecular testings, common molecular techniques. We also look at common molecular techniques. Okay. Later on, we'll be looking at automation in hematology, lymphocyte transformation test, Paul Bonnell test, electrophoresis. So this is the aspect of 521 we are going to cover in this lecture. Okay. So today we are going to do a sort of introductory aspect of molecular analysis with specific interest to hematology. So today we are going to have an introductory lecture on molecular diagnostics in hematology. So we're going to look at a brief history of molecular analysis, understand the concept of molecular diagnostics, understand the functions of molecular diagnostics, understand key molecular techniques, and also the areas of application. So today, basically, I'm going to introduce the lecture. Then the next time, we are going to go into more technicalities. Okay. Looking at some historical developments in molecular diagnostics, we can see some of the key dates. Some scientists like Gregor Mendel, law of hereditary, okay, purification of DNA by Michel, sickle cell anemia mutation was first studied in 1949. You can see that the study of hematological disorder gave the was in front line in molecular diagnosis. So before they start describing the DNA structure by Watson and Crick, who described the structure of DNA in 1953, talk about this, followed by the recombinant DNA technology, DNA sequencing in 1977, in vitro amplification of DNA, that's the PCR, and in 2001, the Genome Project was kicked off. So these are some of the key dates in major discovery that helped in molecular analysis. So molecular analysis, molecular biological techniques, basically they enhance the ability to predict, okay, or identify the number of diseases in clinical laboratory. Okay, they also enable scientists or clinicians to monitor disease progression during treatments. Molecular analysis also help them make accurate prognosis and predict the response to therapeutics. Okay, these are some of the key roles or importance of molecular diagnostics. Okay, and also the short interval required to perform molecular diagnostic tests and analyze their results is also an additional positive aspect of this type of investigation, which results in more efficient patient management, especially in cases of infection where there is need for timely intervention. Okay, we can also have the 5 main areas of hematopathologic molecular testing include the detection of mutation, gene rearrangement, chromosomal abnormalities for diagnosis and prognosis of hematological malignancies. So we'll see some of these importance or key roles of molecular diagnostics as we go on. So, but what is the molecular diagnosis? From our slide here, we can find out that from this our slide, you can see that molecular diagnostics can be referred to nucleic acid-based diagnosis of disorders. Nucleic acid-based diagnosis of disorders. It's a platform where the specimen, the sample for investigation is the DNA. It's an investigation format where we use the DNA to do the investigation. Unlike our normal investigation where we use plasma or serum or the blood, we use the fluid portion of the blood, we use some components of the blood. But here, investigation at the molecular level entails using the DNA, the RNA, the nucleic acid to study a disease or to study monitor therapy or prognosis. Okay, so it refers to nucleic acid based diagnosis of disease, that is the use of DNA and or RNA samples to facilitate detection, diagnosis, subclassification, prognosis, and monitoring of response to therapy. That is how Patterson's described it. Molecular pathology can be also defined as the use of genetic data. These are different views, different viewpoints of some scientists in trying to define molecular diagnosis. Some call it molecular diagnostics. Some can use the term molecular pathology. Okay, so these are some of the views, but they are all main considering the same main starting material, using the nucleic acid to investigate a disorder, to diagnose a disorder, to uh, determine the prognosis, or to monitor the therapy. So molecular pathology, as defined by this scientist, define it as the use of genetic data in addition to standard pathological parameters, okay, to optimize diagnosis and to indicate treatment and prognosis. So this scientist, Tom Wilson, added in his definition that the genetic data must be correlated also with the standard pathological parameters. You don't just do genetic data evaluation in isolation to the standard pathological parameter, that is using the serum or other body fluids. That is what he's trying to bring up in his own definition of molecular pathology. Then another view of molecular pathology by Coleman W.B., that molecular pathology represents the application of the principles of basic molecular biology to the investigation of human disease processes. So these are some of the views of the scientists that have tried to define molecular analysis, molecular diagnostics, molecular pathology. Okay, so molecular analysis in hematology. Okay, we can find that DNA analysis is playing an increasing important role in redefining hematological diagnosis and determining the treatment. So over recent years, our understanding of the molecular basis of both inherited and acquired hematological disorder has improved considerably. There are several ways in which this knowledge is now being applied in diagnostics hematology, and some of the areas, some of the diseases where we can employ molecular analysis or molecular diagnostics include for diagnosis, okay, malignancies in which molecular methods are performed for diagnosis and monitoring of minimal residual disease include inacute leukemia, myeloid, lymphoblastic, myeloproliferative neoplasms, chronic myelogenous leukemia, polycythemia vera, essential thrombocytopenia, primary myelofibrosis, myelodysplastic syndrome, mature lymphoid neoplasm, chronic lymphoid leukemia, and lymphomas. These are situations where we can use molecular analysis for the diagnosis of a disorder. We can also use molecular analysis for monitoring of residual disease in acute leukemia. We can also use it to monitor uh, minimal residual disease in chronic myelogenous leukemia, the quantification of fusion messenger RNA transcript due to what? Translocation, and so many other disorders. We can also use molecular analysis to study also inherited hematologic disorders. Some inherited hematologic disorders can also be investigated with molecular methods, such as when we have a erythrocyte disorder, okay, the hemoglobinopathies like the sickle cell disease, membrane abnormalities, have enzyme deficiencies, erythropoietic porphyrins, leukocyte disorders, quantitative disorders, functional disorders, coagulopathies, thrombophilia, bone marrow failure, quantitative disorder, platelet disorder, storage disorder. In some of these disorders, we employ molecular analysis. So there's a whole lot of cases, situations in hematology, even in pharmacogenetic testing for genetic variants affecting therapy, okay? We can also employ molecular techniques. For example, patients that are taking warfarin, okay, we can use molecular analysis to investigate the sensitivity of warfarin, okay? So many other drug monitoring like the ABL1 gene mutation, okay, imatinib, also in patients that are taking imatinib resistance can also be monitored. Pharmacogenic testing for genetic variations, okay. So there's a whole lot of conditions and situations, disorders, where investigating the DNA molecular analysis can help us in hematology. It has a very versatile, wide applications. So that is why we are studying it. So history of molecular diagnostics, we say that using sickle cell as a case study, okay, it's a genetic disease caused by an A to T point mutation in the cis AA beta chain of hemoglobin, which produces the S, which polymerizes in the deoxygenated state, resulting in physical deformation or sickling of the red cell. Red cell will change from its biconcave shape to a sickle shape. Okay, then sickle erythrocytes, they promote vaso-occlusion and hemolysis, which are two major hallmarks of the disease. These cells, red cells, are now sickled, and the sickle shape does not allow them to move freely inside the blood vessels, okay, allowing them to be easily deformed and hemolyzed, okay. Linus Pauling introduced the term molecular disease in the medical vocabulary based on their discovery that a single AA change leads to sickle cell anemia. In principle, their findings set the foundation for molecular diagnosis. So we also we'll still go into details about the DNA structure. We are just introducing molecular diagnosis, the importance, the areas where it can be applied, and all that in this section of the lecture. So the discovery of the DNA structure, as you will see later and as we know, by J. Watson and F. Crick, okay. The structure of DNA was published in Nature, where they stated that they wish to suggest a structure for the salt of DNA. This structure has novel features which are of considerable biological interest. This is one of the most important biological discovery the 20th century, the discovery of the DNA structure by J. Watson and Crick set the stage for molecular analysis, investigations, diagnostics, whichever way you want to describe it. So discovery of the DNA structure continued in 1955. 12 scientists received Nobel Prizes. 1958, Frederick Sanger worked on insulin. 1962, J. Watson, F. Crick, and Wilkins describe the DNA structure. 1980, Frederick Sanger came up with DNA sequencing process where we try to describe the genetic sequence, the genetic sequence, the codes that makes up a gene was sequenced. Then Caesar Milton and others, they worked with what? Monoclonal antibodies. So these are some of the hallmarks, some of the scientists, and the key dates, the key findings, the key milestone scientific discovery that enhance or led to molecular investigations. So the first seed of molecular diagnostics were provided in the early days of recombinant DNA technology, called cDNA cloning and sequencing provided the basic knowledge on the primary sequences of various DNA. This is the process that involves molecular analysis, cDNA sequencing, cDNA cloning, and sequencing. Okay, this is the molecular technique. It's a technique in molecular analysis, cDNA cloning, and sequencing. They are all molecular techniques that are employed in the study of the DNA sequence. Okay, this cDNA cloning and sequencing provided the basic knowledge on the primary sequence of various genes. They were able to know the sequence, okay, the makeup of so many genes by this process of cloning and sequencing. DNA sequencing provides several probes, allowing the analysis via Southern blotting of genomic regions. These are molecular techniques. When you hear about Southern blotting, it's a molecular technique they use to identify some concept and application. Helped in restriction fragment length polymorphism. Another molecular technique. Don't bother about the technique. We'll try to describe some of them when we go into describing some technological techniques in molecular analysis. So they also track a mutant allele from heterozygous parents to a high-risk pregnancy using molecular techniques. So this is Karimulis, who invented the PCR, the polymerase chain reaction. And he received the Nobel Prize for this discovery in 1933 because his discovery helped to study the gene structure, the DNA, the codes, the genetic sequence, the genetic code. The PCR, he discovered the PCR that enabled the scientists to now amplify, produce more of this DNA so that they will be able to study it. So the unusual origin of PCR. I also urge you to go and read how he came about this process of polymerase chain reaction. So PCR revolution. So the discovery of PCR, having established the DNA sequence by Crick, okay, Kalimulis was able now to develop this process of PCR polymerase chain reaction, whereby he was able to produce a large amount of DNA molecules, which they used in the investigation or in study of any disorder or disease. So PCR has greatly facilitated and revolutionized molecular diagnosis. First most powerful feature is that a large amount of copies of the target sequence generated by its exponent amplification, which allows the identification of a known mutation within a single gene, a single day, sorry. So the process of PCR helped scientists to study the DNA, to study the DNA of a gene. Okay, it helped them in such a way that they were able to multiply the amount of DNA, that is, increase the quantity so that they'll be able to subject it to molecular analysis. Okay. So also the US government started this human genome project after PCR methods and other molecular techniques came on board. The US government project, coordinated by the Department of Energy and National Institute of Health. The goal of this human genome project is to identify all the genes in human DNA, and they have achieved that. All the genes in human DNA has been identified. They also to determine the sequences of 3 billion bases that make up human DNA, the sequences that make up the human DNA. And then they create a database, that's a database of this human DNA, in such a way that if there is any disorder that came up, they'll be able to match it to that data and see where the problem is. Then develop tools for data analysis. Then address ethical, legal, and social issues that arise from genome research. This is what the Human Genome Project by the US government set out to achieve after the discovery of molecular techniques, molecular methods, lasted from 1990 to 2006, and it was, of course, successful. So advantages of molecular diagnostics. We'll look at its impacts on human disease, novelty, that is discovering something newly. So it has there are some advantages of molecular diagnostics when we look at its impact on human disease. Let's talk about novelty. Okay, the discovery of potential novel molecular markers of human diseases. Okay, identification of novel molecular markers of human diseases, utility of molecular markers to develop useful molecular assay for the detection, diagnosis, and prediction of diseases. Just like I was saying earlier, the areas where we can employ all this molecular analysis in identifying some diseases. I was saying earlier that the molecular biology technique enhance the diagnostics ability to predict or identify increasing number of diseases in the clinical laboratory, enabling clinicians, these are the advantages of it, to monitor disease progression during treatment and make accurate prognosis. Like, for example, we can use it in diagnosis of leukemia. We can use it in diagnosis of polycythemia vera, essential thrombocytopenia. These are hematologic disorders. We can also use it to monitor diseases, so that we can monitor minimal residual disease in acute leukemia. Okay, you can also use it to monitor disease in chronic myelogenous leukemia. Okay, it can also be used to identify inherited hematologic disorders. You can use also molecular diagnostics to identify inherited molecular diagnostic disorders, such as the hemoglobinopathies, okay, coagulopathies, thrombophilia, platelet disorder, storage disorder, and all those hematological disorder. This is its advantage on impact in human diseases. We can also use it to monitor disease accurately. It allows for better prediction of response to disease treatment and treatment response to treatment of and disease outcome. Okay, reliable is a reliable and accurate diagnosis of disease. The molecular analysis is more precise, more accurate, at least, than other traditional analytical procedures. Okay, it also gives proper subclassification of diseases. Okay, it helps to determine the most appropriate treatment, personalized medicine, most appropriate medical treatment, fitting dosage and combination of drug based on the patient genetic background. This molecular diagnostics, molecular analysis, molecular investigation, or whatever term you may choose to describe it with, help in this aspect, especially in trying to use an individual's genetic makeup, because some individual may be resistant to a particular drug while some person may not be resistant to that particular drugs. So when the genetic sequence of an individual is known, it will help the clinicians to give the particular drug or particular treatment that that individual will not be resistant to. Your treatment will be tailored with depending on the type of genetic makeup an individual has. So this is a very good advantage for molecular analysis. It reduces patient morbidity and mortality too. Okay, impact on human disease, practical application, that is in diagnostics, in prognosis, outcome of a disease, to predict the outcome of a disease. There are some persons, knowing their genetic makeup will help you to predict that treating this patient may respond well to treatment. This patient may not respond well to this treatment depending on their genetic makeup. It will also help you to predict the possibility of looking at their genetic makeup. Can also help you to predict that this person with this genetic makeup is predisposed to having this disorder. Okay, these are some of the things you can be able to say or come up with, knowing someone's genetic makeup. You can be able to say, this person suffering from this disorder may respond well to treatment. There are some genetic variation that when somebody has displays it in his genetic makeup, it will help the clinicians or scientists to say this person have a better chance of responding well to treatment or not, or that this person having this thing is exposed to coming down with such a disorder, and also therapeutic response to disease treatment. Okay, I think I've explained that earlier. So impact on human disease. Molecular pathology has broad applications, has very broad applications in hematology, infectious disease. It cuts across all area of science. There's no area of science, any area of science where you can get a genetic material. Anywhere where you can get the genetic material, where you can get the DNA. Of course, you know, the DNA contains the nucleus. The mature red blood cells, as you know, does not contain the nucleus. So anywhere you can get the cell that has DNA, nucleic acid, can be subjected to molecular analysis, molecular testing, molecular investigation. So it cuts across not only hematology, goes into microbiology, okay, infectious diseases, solid tumors, genetic diseases, identity testing, paternity testing. So molecular analysis, molecular pathology, molecular diagnostics, whatever term you use to define it, cuts across so many aspects of human disease. So impact on human disease, we'll take it as some specific examples of what we have been saying earlier. In single gene disorder, we have the cystic fibrosis, hemochromatosis, Stark's disease, and sickle cell anemia. Here, molecular investigation help to identify the culprits, the genetic change, the genetic mutations that actually led to the presentation we see in the individuals. Then we have in polygenic disorder. These are disorders arise due to mutation in more than one gene, like the cleft lip, cleft palate, constricted stomach, etc. Common disease like diabetes, blood pressure, heart problem, and asthma. Okay. Chromosomal disorders like Down syndrome, Fragile X syndrome, Klinefelter syndrome, Triple X syndrome, Turner syndrome, trisomy 18, trisomy 13. So these are some of the single gene disorders, okay, and polygenic genetic disorders where molecular investigation can actually be precisely used to identify the um, disease or the condition. So it also help in discovery of potential novel molecular markers of human disease, identification of novel molecular markers of human disease, utility of molecular markers to develop useful molecular assays for the detection, diagnosis, and prediction of disease. Okay, identity testing, paternity, that's parentage, clinical testing. Okay, it can be used for identity testing to determine the parentage, or in clinical testing to investigate an inherited disorder or any other disorder that expresses itself at the molecular level. Infectious diseases, okay, qualitative and quantitative detection of infectious agents, microbial identity testing, genotyping and drug resistance testing. Molecular analysis investigations are applicable in all these areas of investigation. Okay, in molecular oncology, can use it in diagnostic testing, disease prognosis, determination of predisposition, like in all these malignancies, leukemias. You can use it in hemopathology, diagnostic testing, determination of clonality, identity testing, parentage clinical testing, infectious diseases, qualitative and quantitative detection of infectious agents, microbial identity testing, type and drug resistance. I think it's the same part. So in the next lecture, I think we'll now go into the structure and the structure and function of the DNA. I told you that we'll use this time around to just introduce the course and then tell us the importance and significance of studying molecular investigations, the history surrounding it, and just a bit of introduction today. So if you have any questions, you can ask